Welcome back with a new lecture. In this lecture, we will discuss about the cell membrane structure, the transport across cell membrane, its function in detail. So first, we will discuss about the introductory remarks about plasma membrane. Plasma membrane or cell membrane, it is also known as cytoplasmic membrane, is a biological membrane that separates interior of all cell from the outside. So cell membrane basically act as a protective layer for the interior of the cell, and it separates it from outside environment. The basic function of cell membrane is to protect cells from its surrounding. The structure. Cell membrane is a delicate two-layered structure of lipids and proteins. The most widely accepted structure model of membrane is fluid mosaic model, which was uh, shown by Singer and Nicolson in 1972. The double layer of membrane consists of lipid layer made up of a hydrophilic polar head along with two hydrophobic non-polar tails. So we can see here in this figure the double layer of membrane, the visible double layer of membrane is made of lipid layer. You can see here, this is one lipid layer, this is another lipid layer. So it consists of two parts. The head part, which is hydrophilic water soluble, also known as polar head, and the tail part is hydrophobic non-polar tails. So it made a lipid layers, and this is the water environment. So lipid arrange themselves in a double layer structure with hydrophobic tails pointing inward and hydrophilic heads facing outwards. This two-layer structure is known as a lipid bilayer. So we already see that in a membrane structure, two lipid molecules arrange itself in such position that the heads are facing outwards and the tails are facing inwards, and this make a structure known as lipid bilayer because it consists of two lipids molecules. There are three main types of proteins beside lipids. Cell membrane also contains three types of proteins. First one is integral proteins. These are permanently present inside the bilayer, and they can only be separated from the bilayer with the use of detergent. So integral protein are permanently present inside the lipid bilayer. Next is peripheral proteins. They are only on the exterior of the membrane. These proteins adhere only temporarily to the membrane by attaching to the integral membrane. Proteins are penetrating to the outer regions of the membrane. So peripheral proteins are adhere temporarily to the cell membrane. Next is channel proteins. It is found in the membrane and are needed for the movement of molecule across membrane. So channels proteins are present inside the membranes, which helps in the movement of molecule across membrane. Plasma membrane also contains carbohydrates, which are either attached to proteins or lipids, known as glycoproteins and glycolipids. Cholesterols are also found in the cell membranes, which affects the fluidity of cell membrane. So you can see here, this is a detailed structure of cell membrane. This is cytoplasm region. This is extracellular fluid. So you can see the reddish area is known as consist of phospholipid bilayer. You can see this is one lipid layer, this is another lipid layer. So the reddish color is, this is the heads, These reddish balls are the heads, these are the tails, and these is integral proteins, which is globular proteins. These are the proteins channels which transport molecules. These are the glycoproteins, proteins when protein combined with carbohydrates. These are the glycolipids. These are the cholesterol. This is the peripheral protein which temporarily attached to the cell membrane, and these are the integral proteins. So these are the different types of lipids, proteins, carbohydrates, and cholesterol found in the cell membrane. Next, the important topic is the movement across cell membrane. So the movement of substances across membrane can either be passive, which do not require the input of cellular energy, or active, which require energy. So two types of processes occur: passive, which do not require the expenditure of energy, and active, which require energy to occur. Passive transport consists of diffusion, osmosis, and facilitated diffusion. Diffusion is a process in which molecules move from the area of higher concentration to the area of lower concentration. Diffusion transport material across membrane and within cells. So you can see here, this is cell membranes, this is the extracellular fluid, this is the cytoplasm. So here the concentration of molecule is higher. So from here, diffusion of these molecules will occur from this higher region to this lower region. So this is simple diffusion. Next is osmosis. Osmosis is the movement of water across the membrane from the area of low solute or high solvent concentration to an area of high solute or low solvent concentration. Osmosis transport only water across a cell membrane. The semi-permeable membrane limits the diffusion of solutes in the water. So osmosis only transport water molecule, because the cell membrane, we know that is semi-permeable, it limits the diffusion of solutes present inside the water. Aquaporetes proteins, which is also known as water channels, it facilitate or transport water movement, and they play a large role in the process of osmosis. These are present in the cell membranes. Next is the facilitated diffusion. It is the process by which molecules are transported across the plasma membrane with the help of membrane proteins. So in facilitated diffusion, a membrane protein is involved. Integral protein are used in facilitated diffusion, which are also known as transport protein. They function either channels or carriers. So for facilitated diffusion, the membrane protein which is used is known as integral proteins, and these are also known as transport protein, and they may act either a channel or a carrier to carry something, or channel or to allow something to go out. So you can see here, this is a protein, this is lipid bilayer plasma membrane, this is the protein channel, and it helps to transport certain molecules across membrane. Another type of protein embedded in the plasma membrane is carrier proteins. It binds a substance and trigger a change of its own shape, moving the bound molecule from the outside of the cell to an interior, and depending on the gradient are typically specific for a single molecule substance. Such type of carrier proteins are specific for single type and specific type of substances, and when that substance bind to these carrier proteins, it changes its shape and moves the bond molecule from outside to the inside. So this is the outside, the exterior, and when some molecule specific to this membrane attaches to it, it changes its shapes and allow and open and allow this molecule to come here to the interior. This is carrier protein. So it changes its shapes according to the specific molecule to be transported. The next step is active transport, in which First one is primary active transport. It utilizes energy in the form of ATP to transport molecule across the membrane and against the concentration gradients. Therefore, all groups of ATP-powered pumps contains one or more binding site for ATP because it involves ATP molecules. The important pumps in animal cells is the sodium-potassium pump, also known as sodium-potassium ATPase. This sodium-potassium pump maintains electrochemical gradients, which means that it contains the correct concentration of sodium and potassium inside the cell, inside and outside of the cells. Sodium and potassium pump moves two potassium into the cell and moving 3 sodium out of the cell. Cells use energy to maintain concentration gradient of some ion across the cell membrane. So this concentration gradients and the concentration of different ions across membrane is very necessary, and cell use energy to maintain this concentration gradients. For example, kidney cell uses up to 25% of its ATP per ion transport. Electrically active nerve cell uses 60 to 70% of energy to pump sodium and out and potassium into the cells to maintain the concentration gradients of the membrane. So you can see here, this is the plasma membrane. So this is the sodium. 3 sodium molecule will go outside and two potassium molecule will come inside. So this is the sodium potassium pump, and it will make a concentration gradient, that the concentration of sodium and potassium inside and outside of the cell. Secondary active transport is the transport of molecule across cell membrane utilizing energy in other form than ATP. And the energy comes from the electrochemical gradients produced from the primary active transport. It is made by the primary active transport of sodium out of the cell, creating a higher concentration extracellularly than intracellularly. When sodium is transported outside of the cell, so the concentration of sodium is higher outside than inside. This gradient becomes energy as the excess sodium is tried to diffuse to the interior of the cell, which provide energy needed for the co-transport of other molecule. So we know that sodium is higher outside, so it will come inside of the cell. So this gradient's energy excess sodium when come inside of the cell, it provides energy for other molecule to be transported with sodium. So the example is sodium glucose co-transporter used to transport glucose and sodium back into the cell via antiport. Antiport are those proteins that allow molecules to go in the opposite direction, or symport when both molecules move in the same direction system. So sodium glucose co-transporters transport glucose and sodium at a time. So this is known as secondary molecule, in which one molecule co-transport another molecule with itself. So you can see here, this is the extracellular fluid, this is the intracellular fluid, this is plasma membrane. So this is the sodium. We know that the sodium concentration outside is higher, so this sodium will come inside. So when this come inside, it create a kind of energy which help to enter this glucose molecule with itself. So sodium, along with sodium, this glucose molecule also enter to the cell. So this is known as secondary active transport, and this type of transport occur either symporter or antiporter. Symporter means when the two molecules come in, move in the same direction. You can see A molecule and B come inside the in the same direction. An antiport system, the two molecules goes opposite direction. A come here and B come there. So it transport in opposite direction and it transport in the same direction. So these two systems are involved in the secondary active transport. The next type of active transport is endocytosis. It is the process in which cell absorbs molecule by engulfing them, by eating them. The plasma membrane creates a small deformation inward which is known as invagination, in which the substance to be transported is captured. The deformation then pinches off from the membrane onto the inside of the cell, creating a vesicle containing the captured substance. Endocytosis is either internalizing solid particle, which is known as phagocytosis, or liquid molecule known as pinocytosis. The next type is receptor-mediated endocytosis, RME, also called as clathrin-mediated. It is a process by which cell absorb metabolites, hormones, proteins, viruses, which is strictly mediated by receptor present on the cell surface. So this is receptor mediated endocytosis. And the other two is simply endocytosis, in which one is phagocytosis and the other one is pinocytosis. So next we will discuss about this is endocytosis, in which phagocytosis, pinocytosis, and receptor-mediated cytosis is given. So you can see we already discussed in phagocytosis, this is plasma membrane. In phagocytosis, a solid particle is engulfed, so the plasma membrane become deformed inward. This is invagination. Also pseudopodium, this is invagination and engulfing the solid particle and move inside to the cell. This is phagosome, food vacuole. This is the phagocytosis, in which a solid particle is engulfed by the cell membrane. Next is the pinocytosis. Here the liquid molecule is engulfed by the cell membrane. So this invagination, this is the vesicle formation. The third one of endocytosis is the receptor-mediated endocytosis, in which clathrin receptor-mediated is important. Clathrin is a protein. You can see here, this is the membrane. Here the receptors are present. These are the receptors. This is the clathrin protein, coated pit. This is proteins. So when a virus, proteins or hormone come and bind to the receptor, so the cell invaginate and capture those hormones, viruses, something which they capture and form a vesicle. So these are the different types of active transport, which is known as endocytosis. Now the last one is the function of cell membrane. Cell membrane surrounds the cytoplasm of living cells and physically separating the intracellular components from the extracellular environment. So cell membrane protects the internal environment of the cell from the external environment. So it is very important function of cell membrane. Next is cell membrane plays a role in anchoring the cytoskeleton to provide shape to the cell, and is attaching to the extracellular matrix and other cells to hold them together to form tissues. So the cytoskeletal system inside the cell, the CM play a role to anchor that cytoskeleton to provide proper shape to the cell, and in attaching to the extracellular matrix, and it hold other cell together to form tissues. So it is also a very important function. Cell membrane is selectively permeable and it able to regulate what enter and exits the cell, thus facilitating transport of material needed for survival. So cell membrane act as a selectively permeable membrane and act select what is good for a cell and what's not. So it regulates the transport of material needed for the survival. So these are some important functions of the cell membrane. This is all about cell membrane. Thank you for watching. See you next time.